Hi, guys. All right. So, right now we're going to be talking about DNA, RNA, proteins, the cell cycle, and cell division. So, we're just going to jump right into it. So, DNA is what makes up the chromosomes, the genes that code for your traits. So, think whether you're tall, short, what your hair color is, eye color, whether or not you have freckles, or not you can roll your tongue, whether you have detached earlobes or attached earlobes. All of these things are going to be determined by your DNA and proteins with gene expression. So, nucleotide structure. DNA is a twisted ladder looking type of structure called a double helix, where two strands of genetic material coil around each other, and each strand is made up of a sugar phosphate backbone on the outside with nitrogenous bases on the inside. Our nitrogenous bases are adenine, guanine, thymine, and cytosine in DNA. And that's what makes up the rungs. So all of the bases are going to be stuck to the sticky sugar, and then the phosphate is going to be on the side. That's the phosphate's only attached to sugar. Sugar also attaches the nucleotide base. These three pieces, the phosphate, sugar, and base, are all called a nucleotide structure. I said nucleotide base, I meant nitrogenous base. So a phosphate, a sugar, and a nitrogenous base all form a nucleotide structure. All right, so DNA, deoxyribonucleic acid, double-stranded. Let's get into it. So, like I said, phosphate, sugar, backbone, and a base is the rung. So base, sugar, phosphate. Base, sugar, phosphate, right? Base is stuck to the sugar. So we have a phosphate, a sugar, and a base here. Phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base. Well, a DNA double helix is, think of it as two pencils, one eraser end up, the other one eraser end down, sitting side by side, and that's the way that DNA strands are laid out. So that's called 5 prime. You have a 5 prime end and a 2 prime and a 3 prime end, and we'll look at that in a second. So notice that these nitrogenous bases are different colors. So we just went down one strand. We're going to go back up that other strand in the same way with a base, sugar, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, phosphate, sugar, base, all the way back up, right? So we have 5 prime to 3 prime, 3 prime, 5 prime to 3 prime. So 5 prime up here, 3 prime down here, 5 prime here, 3 prime there, with our rungs in the center. So notice that the nitrogenous bases are different colors. Here are our nitrogenous bases in DNA. We have adenine, which is yellow up here, thymine, which is blue, cytosine, which is purple in this image, and guanine, which is green. Adenine and thymine are always going to pair together. Cytosine and guanine are always going to pair together. That is what will happen every single time. They fit like puzzle pieces. They only fit together one way. And those are going to be your base pairs every single time. So make sure you understand that AT always and CG always. So RNA, ribonucleic acid, it's the same kind of pattern, but it's single-stranded. So what happens here is a different RNA comes in and makes base pairs with the other side, so we don't need another strand. Bases are similar, except there are no thymines in RNA. All of our thymines are replaced with uracil. But same thing here. CG will always pair together, cytosine and guanine. But adenine, instead of pairing with thymine, because it doesn't exist in RNA, pairs with uracil. So AU always and CG always. Please understand that. ATCG in DNA replication, ATCG. But once we start transcribing to RNA, it's going to be AU, uracil replaces that thymine, and CG always. So apples in a tree, car in a garage, easy way to remember. or you can just remember ATCG. All right, so you have these ladder-like structures that coil into a double helix, right? They twist, they twist, they twist, they twist. You can see the increased twisting as you go up and coiling until they coil into this kind of X-shaped chromosome that's held together by a centromere. Centromere in the center holds that chromosome together. That'll be important when we talk about how it breaks apart in mitosis. But that's how we go from a ladder DNA double helix to a chromosome. Lots of twisting, lots of coiling. We're not going to get into the super details of this because we could teach a whole class just on replication, transcription, translation. So when it comes time to make a new cell, DNA has to double down, right? It has to double itself. How do we do this though? So that each cell can get a full set of DNA. So we have to unzip it, right? We have to unzip that DNA. And we have two enzymes that come in that are crucial to DNA replication. These two enzymes that help with this are DNA polymerase and primase. These two enzymes direct the breaking of the bonds, what bases get filled first, and what they're going to get filled with. So DNA polymerase breaks the strand, and primase directs which base goes next. That's all you need to know. You don't need to know a whole lot else. So this DNA strand is getting duplicated, right? It's getting replicated. We're going through DNA replication right now. The blue strand just got unzipped. That's our original strand. And then the orange is going to be the new strand. Original is the blue, it got unzipped. Orange is the new. So remember, we are going in a 5' to 3' direction. The new strand starts at the end and works its way in. So that's 5' to 3', and it can go pretty fast. We call that the leading strand. Now down here, we call this the lagging strand. Remember, if we're going 5' to 3' this direction, we have a complement strand right here going 5' to 3' in this direction. So this one, 5' to 3' left to right. The lagging strand is 5' to 3', but it's going right to left. It's going backwards. This takes longer. The top 5' to 3' is fast. As it unzips, it's just boom, 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 makes pairs. The bottom part is slow. It can only pair in sets of 3, so it has to wait for another DNA triplet to be available. That's what we call sets of 3 nucleotides. We call them DNA triplets. So if you look specifically at the bases, we're going to take an original DNA strand right here. We're going to unzip it. Boom, boom. Here's an original. Here's an original. And we're going to copy it. So there's the new strand. So ATC, TAG, GCT, CGA, GCA, CGT. Be able to do this. Stare at this. Remember, AT always pairs with CG. Be able to make a new strand of DNA from an original strand if I were to ask that of you. Know the base pairs and what bases pair together. Well, if we're going to copy it, we're going to make an mRNA, a messenger RNA, and then we're going to make a protein. That's the whole reason we do all of this, right? And it all starts with the original DNA strand. Again, when we unzip the original DNA strand, bases come in and fill it. Be able to fill in. If this new strand was blank on each side, be able to fill in the triplets, the DNA triplet pairs. Not pairs, but triplets. Make sure you know them. All right, so that's what happens if everything goes right. I don't want you to know very, very specific things about genetic mutations, but I do want you to know what happens in these 3 scenarios. So mutations can happen. Sometimes bases, an extra one can get inserted. Sometimes one can get deleted that should be there. And then sometimes one gets substituted that shouldn't be there. So that should have been a C, substitute with a T. That's not good. Over here, we should have had a T. It's gone. It got deleted. And over here, we got an extra T for Y. Sometimes these mutations don't really lead to much. Other times, they can be fatal. That's all I want you to know about genetic mutations. Again, we can teach a whole class on this material, but we're just going to touch on it. All right, so DNA replication. DNA replicates and then it has to transcribe to RNA, which then translates to proteins. We won't get, again, too in-depth with transcription and translation, but we're going to cover the basics so that you guys understand what's happening. Here we need mRNA to be made. What's mRNA? mRNA is messenger RNA. So remember, mRNA can leave the nucleus with code from DNA to go to the ribosomes with the intention of making proteins that then lead to gene expression, which we're not going to talk about gene expression. So this DNA is sending an order to make a protein. mRNA is going to go to ribosomal RNA, rRNA, within the ribosome, and tRNA is also going to go to that rRNA, and they're going to come together to make a protein together. So we started with one DNA, we split it wide open, and we made a duplicate. In order to make a protein, when we split DNA open, we're bringing RNA nucleotides in instead of those DNA nucleotides. So remember, we don't have any more T's. Now we're AUCG. Got rid of the T's once an RNA comes to pair. So we take our DNA triplets and we make an mRNA with RNA base pairs, AU and CG. When we're talking three letters in DNA, we call it a DNA triplets. In mRNA, we call it a codon. So I got a little ahead of myself. Okay, so here we go. So DNA triplets and mRNA codon. So we're at transcription of mRNA. These mRNA codons have come in to pair with the DNA triplets. All right, so mRNA is the UAG, CGU, CGU. Because no more. Oh, that should be an A. Oh, look at that. There we go. CGA, CGU. So UAG, CGA, CGU. We got rid of the T's with mRNA. All right, so the mRNA is the UAG, CGA, CGU. That single strand of mRNA then leaves the nucleus and it goes to the ribosome where it undergoes translation to a protein. So again, DNA replicates and then it transcribes or rewrites itself into RNA. RNA then translates into a new language called a protein. So you do need to know that. DNA replicates, transcribes to RNA. RNA translates into a new language called a protein. Anyway, so now we're translating that mRNA strand. So we have transfer RNA coming into the ribosome. Each one, there it goes. With each one carrying 3 bases. So each one has to carry the exact right anticodons to match up with the mRNA codons. So UAG with AUC, CGU with GCA, CGU again with GCA. Oh wait, now, well, I fixed it here. That's an A. And that's an A. A U. Okay. Sorry, guys. Save that. Where was I? So UAG, CGA, CGU, AUC, GCU, GCA. And then down here are the proteins or the amino acids linked to those exact anticodons. So on the one end of tRNA anticodons are the nucleotides, on the other end are the amino acids. So anticodon on one, amino acid on the other end. And then notice that the amino acids are in a whole different language. Remember, once we strand amino acids together, we call those proteins. Call them peptide bonds, peptide chains, polypeptide chains, proteins. But anyways, be able to do this. Be able to do replication, be able to transcribe to mRNA, and then RNA to RNA is not going to be bad because you already know that the uracil replaced the thymine. I won't make you learn the amino acids. Okay. If you're, I mean, here's what that looks like. This is a little bit more complicated, but you can kind of see the whole process of mRNA leaving through the nuclear pore and heading its way down to the ribosome, tRNA coming with that exact anticodon match to match up with the mRNA, and then little amino acids getting synthesized. But if it's still confusing to you, this video is a pretty fun little video by the Amoeba Sisters. It's on YouTube. You can Google this as well, and it should simplify the process for you a little bit more with cartoons. But let's talk cell cycle. Okay, so when a cell divides, it has to duplicate its DNA. That is what this is all about. G1 is gap 1 phase. That's where the cell grows until it gets to that point, R, right here, right before S phase, right before synthesis phase, to decide whether it needs to divide or does it need to wait. If there's space, sure, let's divide. Fun fact, cancer cells don't listen and they divide anyway, and that's how tumors happen. Anyway, so gap 1, we grew, we're good to divide. Let's move on to S phase or synthesis phase. So synthesis phase is where we double our DNA. That's where DNA replication happens and really gets us ready for gap 2 phase, where the cell continues to grow and prepares to divide. So here the fibers and the centrioles form to get ready for cell division. Then we're ready for mitosis. So everything before mitosis we call interphase. Interphase takes the longest time. Cell grows, replicates DNA, cell grows some more, and then it's ready to divide. M phase is where mitosis happens. So all of it is interphase until we get to M, which is mitosis. So mitosis only somatic cells go through mitosis. Somatic are the body cells. Our gametes or sex cells, they actually go through meiosis, which is similar, but it's not the same. And we'll talk more about that in A&P2, where we're talking about the reproductive system, the male and female reproductive system. Here we're only talking about body cells and mitosis. So again, this is M phase zoomed in. Everything else up to that point was interphase, right? So here we are, mitosis. So PMAT, remember, don't worry about prometaphase. We're not talking about prometaphase. We're talking about prophase, metaphase, anaphase, telophase, and cytokinesis. PMAT, prophase, metaphase, anaphase, telophase, and cytokinesis. Just remember PMAT and then cytokinesis. So during prophase, the genetic information pairs up into condensed chromosomes, and then the spindle fibers show up. Spindle fibers are what those chromosomes are going to align on in metaphase, and they're going to go towards the center of the cell. So prophase, genetic info pairs up into condensed chromosomes, spindle fibers appear. Metaphase, middle, chromosomes line up in the middle. Anaphase, I always remember ANA. So ANA, put the A's on the poles. The centromere breaks up here, and then the sister chromatids start pulling off to the side. So the chromosome breaks apart because the centromere breaks apart, and then the spindle fibers are pulling the sister chromatids to opposite poles. So we can call the poles A and A. So remember, Anna, they're getting pulled apart to the poles. Telophase, a nuclear membrane reforms around those two sister chromatids that got pulled apart. So all that's left is that the two cells have to be pulled apart, right? So that's where cytokinesis comes into play. Cyto-cell kinesis movement, the cells are moving apart. And boom, we have two new cells. Each of these two new cells is the exact same as the original cell. Both of those cells will individually go through interphase and mitosis until we get a stop signal that we've made enough cells. If that doesn't make sense, please let me know. But that's it. That's our lecture on cell function and genes.